Looking back at this original example of linked genes where A1 and B1 are on the same chromosome, remember, this is the P0 or parental generation. These are their gamete haplotypes. A1 with B1 has to be a gamete that's produced by this parent, and A2, B2 from that parent's genotype. And those produce an F1 individual that's heterozygous, A1 and A2, B1 and B2. And a geneticist might be asking, for example, how far apart are those A and B genes on the chromosome? So we want to be able to measure how frequently the A1 and B1 alleles are separated from each other during recombination, and likewise the same question for A2 and B2. That is, how often do we get A1 with B2 and A2 with B1? In this generation, the way we conduct this experiment is to cross the F1 dihybrid, this individual, with a tester individual. And we've already seen what a tester genotype is. It's an individual that's homozygous for one set of alleles. So in this case, they're homozygous for A1 and they're homozygous for B1. So again, what's going to happen is, at the tetrad stage of meiosis after synthesis, we're going to have A1s and B1s and A2s and B2s lined up in a tetrad, and recombination will occur somewhere randomly. It might be between these genes, it might not be between these genes, A and B. The same thing is happening over here in this tester individual, but the difference is that because they're homozygous, there's only A1s and B1s. Whether or not recombination happens between these chromosomes or not, we get the same F1 gamete haplotypes. So 100%, and this is why we use this tester, is because we know for sure the gametes of this individual. They're all going to be, because this individual is homozygous at both loci, we guarantee 100% of their gametes, sperm or eggs, are going to be the A1B1 type. So that when we cross these individuals in the F1 generation together to produce the F2 generation, we'll know exactly what the gamete haplotypes were from the F1 dihybrid. And that's essentially the question we're asking when we do any sort of genetic mapping experiment, is what are the individual haplotypes of the gametes produced by a dihybrid individual? So we're going to get out of this cross between these two F1 individuals, the dihybrid and the tester, a number of different genotypes. We'll get, in every individual, we know that they will have one gamete that was A1 over B1. And then the question is, what alleles come from these parents? Half are A1 and half are A2. So we're going to have some A1 and some A2. Those come from this parent. We know this parent only contributes A1s. And then we have to think about what's happening at the B locus as well. So we'll have an equal number, presumably, of gametes from this side that have B1s. And A2 was initially with B2. But if recombination happens, then we will get some A1s with some B2s and some A2s with some B1s, as would happen if this recombination event I just sketched occurs. And we know that from this parent, every individual is going to have the A1, B1 genotype. So let's say you were a geneticist and you were actually doing this experiment and you crossed these F1 parents together and you collected a number of F2 individuals with these types of genotypes. I'm going to refer to my cheat sheet. So let's say you collect 24 individuals with this genotype and 18 individuals with this genotype, and 22 individuals with that genotype, and 36 individuals with the second genotype. This is all the information we need to calculate genetic distance. Here's how. What we need to know, again, is we have certain types of gametes. A1 with B1 was a gamete type that was present in the parent's gametes. That's a parental type combination, A1 with B1. A2 with B2 was also a parental type. And if we see any A1s with B2s that would happen if recombination happens between these two genes, those would be recombinant types, and likewise, A2s with B1s would be recombinant types. So what we need to do, the next step after we find the genotypes of the F2 offspring, is to figure out which of these 4 genotypes are the parent types, P, and which 2 are the recombinant types, the Rs. Now, there's a lot of genetic information down here. The reason that we use the tester, again, is that we know its genotype because it's homozygous. We know that every single one of these organisms, regardless of all of these notations that are going on down here, one A1 and one B1 came from this parent in the cross. So we can cross out, because we know the origin of those alleles, we can cross those out. We don't have to think about those anymore. The haplotypes that are left in the genotypes of the F2s then are guaranteed the gamete haplotypes that produced these individuals in the F2 generation. So we know that we had 24 fertilizations happen with a gamete from this F1 dihybrid that was A1B1, and we had 36 gametes that were A2B2, 18 that were A1B2, and 22 that were A2B1. Now, parent types, parental or recombinant. A1 with B1 is a parental type, so we had 24 parental types. A2B2 is the other parental type. And A1 with B2 and A2 with B1 were the recombinant types if recombination, or when recombination occurs between those two genes. To complete calculating the genetic distance, we take the number of observed gametes that were recombinant types, and we divide that, those are the number of recombinants again, by the total number of individuals. And if I did my math right, that's 100. So this will make the math pretty easy. So in this case, 18 plus 22 is 40, divided by 100 is genetic distance of 0.4. Genetic distance is measured using, unfortunately for you, 3 different types of units. 0.4 is called the recombination fraction, or RF, because that's just what we calculated, the fraction of gametes that are recombinant types. So it's going to be an expression that's easily converted to a percentage. So you could say 0.4 recombination fraction is 40% of gametes were recombinant types, right? 40 out of 100 in this case. The percentage of recombinants, or if you multiply the recombination fraction by 100, that gives you map units, or MU. So in this case, recombination fraction of 0.4 gives us 40 map units. So that's recombination fraction 0.4. Map units, 40. And most confusingly, or maybe unconfusingly, after the geneticist Thomas Hunt Morgan, who was the first person to play around with recombination, actually one of his undergraduate students was one of the ones that first worked all of this out, a map unit is equal to 1 centimorgan. So this is either 40 map units or could also be reported as 40 centimorgans distance between gene A and gene B. So I'd like to leave you now with a couple of questions to think about to bring with you to next class. Here we just calculated the distance between genes A and B as a genetic distance of recombination fraction 0.4, or 40 centimorgans. What if the distance between genes B and C is 0.1, So draw me a genetic map, just a representation of the relative locations of the 3 genes, assume they're on the same chromosome, so that the genetic distance between A and B is 0.4 and between B and C is 0.1. An important point here before you attempt this is to know that genetic distances are additive. So you can add together distances just fine. The second question is, what are the maximum and minimum genetic distances? If genes are at the opposite ends of the chromosome, what is the genetic distance between them? And when genes are exactly next to each other on the chromosome, so that recombination will never ever happen between them, what's their genetic distance?